Well, thanks very much, and thanks for the opportunity to speak here. It's really been stimulating so far to see the talks. What I'm going to be talking about today is a relatively new direction for our group, but we've really found it to be fascinating, and there are lots of unanswered questions that only recently have been able to be addressed owing to the technological advances. So first of all, since I realize most of you don't spend all your day thinking about the human mitochondrial genome like we do, I thought I'd get us all on the same page. And just for those of you who aren't that familiar with this aspect of human chromosomes, I would just quickly give a little bit of background. So, the mitochondrial genome depicted here, of course, is circular, densely packed with genes. There's very little non-coding space, 13 protein coding genes, almost aligned back to back with interspersed with some of the tRNAs, which means, of course, it has its own translational machinery and its own genetic code, as was alluded to before. Furthermore, it's got its own ribosomal RNAs. And the important thing when one tries to tease apart what's going on with regard to cancer in particular is that the genome is present at a huge number of copies per cell. And this number of copies varies greatly from tissue to tissue, even in normal tissues, not necessarily in cancer tissues. And this has implications when one starts to look for analogs of things like loss of heterozygosity. So, of course, in the nuclear genome, you've got loss of heterozygosity events where you lose a piece of one of the chromosomes, and therefore the cell becomes homozygous at that locus. Whereas in the mitochondrial genome, many of the normal cells, in fact, some think all of the normal cells, are so-called heteroplasmic in nature. That is, there's more than one mitochondrial haplotype in the individual cell. So you'll see these dramatic shifts even within tissues from the same individual, and these shifts become even more drastic when you start looking at the cancer genome. Furthermore, the mutation rate is much higher in the mitochondrial genome, again, both in normal tissues and in tumor tissues. So there's a lot of opportunity for selection to act. And I should just, by way of nomenclature, because it's going to come up later, the two DNA strands are typically, sort of classically referred to as the heavy strand and the light strand. And the heavy is actually the reference genome. So what this means is that each cell sort of has its individual constellations within an individual of relative haplotype abundance. And this process of the segregation of the different haplotypes actually starts with this individual's mother when she's in utero, in a sense, because all of her oocytes, when she's just a fetus, are drawn, the mitochondrial haplotypes within them are all drawn from the same pool of her mother's mitochondrial haplotypes. However, they're sort of distributed in a semi-stochastic manner across all the different oocytes. Now, one of these is fertilized, which actually induces a further shift owing to the extrusion of the different polar bodies. And then with each cell division, of course, there's an opportunity for segregation to occur so that when this individual is fully formed adult human, from tissue to tissue, you, of course, have different haplotype abundances. And then in tumor, we see an elevated rate of somatic mutations, which gives rise, of course, to still further haplotypes. Now, there have been classically many diseases where specific mitochondrial mutations have been associated with those diseases. Sort of the better studied of these is so-called MELAS, which is an acronym encompassing all the different phenotypes there. And the interesting thing about this disease is that it's associated with this very specific tRNA mutation. And the severity of the disease is intimately tied to the per cell abundance with respect to the wild type of the mutant allele as compared to the wild type allele. So if the mutant allele is low enough, then there's absolutely no phenotype. And then on the other end of the spectrum, you don't observe any living individuals with a super high abundance of the mutant allele because they're perinatally lethal. And of course, what we want to talk about here is cancer. And specific mitochondrial DNA mutations' role in cancer continues to be quite controversial. There have been many, many papers over the last 10 to 15 years, and more as the sequencing technologies have advanced, that have shown or have demonstrated some sort of association between cancer and different sorts of mutations in mitochondrial DNA. Now, there's still a lot of debate as to whether these mutations are actually causal or they're just bystander effects. But one can start to look at these with the new technologies, and one of the ways to address this question is to see if there's any sort of signal of selection for any of these particular mutations, which could be indicative that they're in fact functional. So any next generation sequencing experiment that one does, whether it's RNA-seq, ChIP-seq, DNA-seq, you wind up just owing to the huge per cell abundance of the mitochondrial genome, lots and lots of mitochondrial reads. And most researchers will just throw them out as sort of nuisance. So what we've kind of been taking it upon ourselves to do is to sort of pick through the garbage, as it were, to see what we can infer from these reads. And what I'm going to focus on today, and as much time as I have, sort of running out now, these sorts of lesions that one can pick up. So one can look at somatic point mutations in indels. One can gauge these heteroplasmy shifts if you look at the matched normal tissue and compare it to the tumor tissue. And you can, by using the nuclear genome as sort of an internal calibrator, one can measure the per cell copy number changes as one compares the normal to the tumor. Now, really what's been holding us back the most is the upstream data analysis. And in fact, many of the results I'm going to present today are kind of preliminary because we're still actually trying to tease out some of the upstream artifacts when one takes these reads that are putatively from the mitochondrial genome and tries to infer these different kinds of somatic changes from them. So our primary data source for this has been the Cancer Genome Atlas, which many of you are probably familiar with, where, owing to this effort, we have literally thousands of matched tumor normal pairs where we can infer all these sorts of lesions from the next generation sequencing. They cover a variety of tumor types, which are these are the TCGA abbreviations, and these are the tumor types that we primarily made use of. And we began this a while ago just as a pilot project where we just grabbed what was available at the moment, and that was just happened to be about 100 breast cancer genomes. And we did this study. This was my former student, Sarah McMahon, who's subsequently moved on to UCSF. We did this study just trying to empirically look at what we could conclude from just examining these mitochondrial mutational patterns. And what we found is that about 3/4 of these patients did carry some kind of tumor-specific mutation. The most we found was 5. And we were also able to, as I said before, gauge the heteroplasmic allele shifts. Now, that 5 mutations in an individual person does not sound like a lot until you realize, the mitochondrial genome is so small that on, per megabase or per kilobase, rate per kilobase measure, it's actually a pretty high level of mutation, especially as compared to the nuclear genome. So these 2 left panels were from a pair of papers that were published a couple of years ago that got a lot of attention in Nature, where they examined the mutation rate across various tumor types, again in the nuclear genome, and just using their metric, the number of mutations per megabase. And they found that, perhaps unsurprisingly, the cancers that were due to exogenous exposures like smoking or UV light had the highest rates of mutation. But if we take just our data from our 99 breast cancer genomes and use the same metric, it's actually higher. This is the median here. It's actually much higher, an order of magnitude higher than even the highest mutators in the nuclear genome. And in fact, breast in the nuclear genome is sort of on the low end as well. So the mutation rate, is definitely, in that sense, quite high. Furthermore, when we broke down these mutations by gene, we found that the, relative to the size of the gene and relative to its sort of inherited polymorphism burden. We found that some of the genes, in particular ND5, seem to have an elevated rate. And when we collapsed them by complex, we found that complex 1 seemed to have an elevated somatic rate as compared to the other complexes. Furthermore, when we looked more closely at the control region, so this is the non-coding region that carries the majority of the regulatory elements, we found that the regulatory elements, in fact, were enriched for these somatic mutations in a statistically significant manner. Even though we were probably somewhat underpowered, we still were able to detect this kind of enrichment. We also found, when we started to break down synonymous versus non-synonymous, that at least as compared to the germline variants, there seemed to be relaxed selection, at least, and perhaps even positive selection for the somatic mutations, where the rate of synonymous versus non-synonymous mutations was, the rate of the non-synonymous versus synonymous mutations was much higher for the somatic mutations as compared to the germline variants. So the genome is perhaps, again, unsurprisingly, much more tolerant of these mutations when they occur somatically, and maybe even select for them. Now, to quantify this a little more rigorously, we, and others have done this, borrowed ideas from population genetics where you just look at the rate of synonymous mutations as sort of the background rate and examine whether the rate of non-synonymous mutations is higher, lower, or about the same. And if it's about the same, then the inference is that it's selectively neutral, that is, perturbations in the gene in this context aren't preferred or selected against in cancer. And the ratio being larger than 1 is deemed to be evidence of positive selection, less than 1 is negative selection. And traditionally, these statistics, the DNDS statistic, its significance, statistical significance, is evaluated by just comparing to the analytic null distribution. So theoretically, what the distribution of the statistic should be. So when we did this, we found, at least in the germline, that unsurprisingly there was strong, almost universally, selection against non-synonymous mutations. However, in the somatic genome, it tended to be either neutral or maybe even some slight evidence for positive selection. Again, we may have been rather underpowered for this effort. What we found that was kind of interesting was that mutational burden, and this was contrary to what we initially thought would be the case, was associated with better patient outcomes when we started looking at survival. And perhaps this could be the result, this is just speculation, of perhaps the tumor is more compromised when it has a higher mitochondrial burden and therefore more susceptible to treatment. But as I say, that's just speculative. A little bit after we published this paper, another paper came out from a group in the UK where they pointed out that the DNDS statistic was in some sense flawed when doing this kind of analysis for somatic mutations in the mitochondrial genome. And the reason for that is that there are endogenous preferred mutational biases in mutations in the mitochondrial genome owing to biochemical reasons, basically, where there's just this strong preference, and this is true over evolutionary time, and this is also true somatically in very short time in the cancer genome. For example, where G to A and T to C substitutions, this is with regard to the light strand, tend to be preferred. And furthermore, the surrounding base content also plays a role in this regard. So if one controls for that, it turns out that a lot of, when they did the analysis, they realized that a lot of the available synonymous changes that were available in the genome had already been used up, as it were, by the endogenous mutational processes. And therefore, when the somatic mutations began to occur, they had more opportunities, again, given these mutational preferences, for the non-synonymous mutations, therefore elevating the sort of DNDS, the numerator of that statistic. So it looked like there was an elevation of non-synonymous mutations, but that was just because there were more non-synonymous mutations available given the preferred mutational patterns. But in our view, they demonstrated that doing DNDS in the usual way was flawed, but in our opinion, they didn't necessarily show that it was true that there was no selection at all, at least not in a very convincing manner. So my graduate student, Sneha Gran, who's depicted there, has undertaken a more global analysis, expanding from the breast cancer genomes and trying to attack all of these en masse. And I'll just briefly have time to touch on some of the results. As I say, this is somewhat preliminary because we're still sort of weeding out the upstream effects. But if you just look globally at all the mutations and map them to the genome, you can see that it's not at all uniform. And some protein coding genes seem to have an elevated rate of mutation, and in the D loop, in the non-coding region, of course, you have an elevated mutation rate as well. Furthermore, when you start to break it down by tumor type, liver, this LIHC is liver, always seems to have an elevated rate on average as compared to the other tumor types, and it also seems to have its own mutational signature as compared to the others, having a much higher proportion, for example, of A to C substitutions than the others do. One thing to note is that we do have to, this is contrary to the way this analysis is usually done in the nuclear genome, we do have to represent all 12 possible nucleotide substitutions. And the reason for that is we can't collapse the strands, because in the mitochondrial genome, things are very strand specific, whereas in the nuclear genome, an A to G mutation on one strand is really the same as a T to C on the other strand, and therefore they sort of collapse the two strands and only have 6 possibilities. The reason for this is, one of the reasons that we have to treat it separately in the mitochondrial genome is that mitochondrial DNA violates Chargaff's second parity rule, which says that, and this holds true across pretty much all eukaryotes and all bacteria, that if you take either strand of DNA, it doesn't matter which one, in the nuclear genome, the number of A's tends to be the same as the number of T's. So this isn't the same as the Watson-Crick base pairing. Not globally, the number of A's is the same as the number of T's, but in a single strand. And you can see this by just picking a strand and looking at the ratios of A's to T's. And this remains fairly constant in G's to C's. Whereas in the mitochondrial genome, it's wildly violated. For example, on the light strand, there are 2.5 times as many C's as there are G's. So nonetheless, we find that nearly half of all the somatic mutations do hit the G's on the heavy strand, or the light strand, excuse me. So what we have done to try to control for these endogenous mutational processes and still do these DNDS calculations is just to do a straightforward permutation analysis. So the way it works is fairly simple. What you do is you just take whatever you want to look at, whether it's specific tumor type, specific gene, or everything as a whole, compute your DNDS statistic, and whatever it is, and then to assign statistical significance to it, you need to generate a null distribution. To do that, you just do a straightforward permutation analysis where you sort of simulate the endogenous mutational process that you observe by permuting all the mutations to identical initial base sites with identical flanking bases and induce exactly the same mutation. And you do that once, compute the DNDS statistic in the simulated data, and then do that another 1,000, 10,000 times. And that gives you a null distribution of the DNDS statistic, and you can compare your normal to that null distribution and thereby generate a p-value and assign significance to it. So when we've done this, again, this is somewhat preliminary, but we're starting to see signals that perhaps there is selection in a tumor type specific and to some degree gene specific manner. If you just do it globally with all your tumor types and all of your genes, there is no significance. It's completely null. However, when you break it down by tumor types, some show very strong, there's sort of extremes where some show very strong selection against protein perturbations, and some show signs of positive selection. And similarly, in a gene way, although on a sort of much smaller scale, seem to show potentially some signs of positive or negative selection. We're doing similar analyses where we're looking at shifts in heteroplasmy. So conceptually, you might imagine that if there's an allele that's tumor preferred, and if it's present in a heteroplasmic constellation in the normal cell, you might recurrently, if you look across multiple individuals, see that allele rise to prominence in a statistically significant non-random manner in the tumor cell. And so we're developing statistical tests to try to assess this. What we see where you start with, so if you just start with the normal cells, the normal matched tissue, and you look at all of the heteroplasmies, again, perhaps unsurprisingly, one sees an enrichment. When there's a heteroplasmic situation, the reference allele tends to be the dominant allele, which is likely just because the reference allele is usually wild type. Now, when you move to the tumor, so here we're plotting the two. When you move to the tumor, we tend to see it rapidly drift toward homoplasmy, where one of the allele rapidly becomes more dominant than the other. Now, this could just be a consequence of rapid cell division, but one of the things it could also be is one of these alleles could be conferring some sort of selective advantage or disadvantage. So developing these statistical tests has been a bit of a challenge because there's a lot of things to control for and there's a lot of different things one can measure. Perhaps the simplest way to do this would be to just simply count which direction, the number of cases in which the wild type or the reference allele rises and the number of cases in which the alternate allele rises, and assess the statistical significance of that. So we're working on that currently. One of the things that's confounding us a little bit is that the likelihood of one allele, even under the null, the likelihood of one of the alleles rising to prominence is intimately tied with how much you start with in the normal cell. And that is, for example, if the reference allele is at very low abundance in the normal cell, it's less likely to even go up at all than it is if it's at a high level. And to some degree that makes sense, and to some degree, it's a little bit surprising. But one can run simulations that sort of recapitulate this effect. We've also been looking at copy number changes, and one of the things that we tend to see in a lot of tumor types is recurrently the normal cell loses, on average, something like 60% of its mitochondrial genome copies as it moves to normal. There are some intriguing outliers here. For example, this is in colon, where you have a number of patients where the number of mitochondrial copies increases in a dramatic fashion. So we're looking to see if there's something interesting clinically about these individuals. We've also begun to look a little bit at mitochondrial RNA expression, and when one clusters on the 13 protein coding genes expressions, we find that they cluster quite distinctly according to tumor type, except with the exception of breast and lung, which tend to be interwoven. So this is quite suggestive. And when we look at whether genes in certain complexes are upregulated or downregulated relative to the matched normal tissue, we find some reasonably consistent patterns across different tumor types. So the last thing I'll touch on is just sort of where we're headed with all this. And one of the things that we'd like to do is to explore the idea of using these mitochondrial changes as biomarkers. For example, one of the areas of high interest currently clinically is circulating tumor cells at one end and circulating cell-free DNA at the other end. And what we'd like to pursue is something somewhat intermediary, that is circulating cell-free intact mitochondria. So Mitha Gould, who's in my group, has been pursuing the idea of trying to capture these cell-free mitochondria in circulation and sequencing the DNA and seeing if one can actually detect tumor DNA from these circulating mitochondria in patients. So we've been looking at some blood, tumor, and normal tissue trios from individuals and seeing if after this mitochondrial isolation, we can pick up some of these mutations, which even if these mutations are not functional, of course, that doesn't preclude their use as biomarkers, perhaps for early detection or post-treatment monitoring or things like that. And then finally, the reason that I just briefly touched on Cancer Cell Line Encyclopedia as one of our sources, what that will allow us to do is to start to look at functional experiments. If we have candidate mutations that are present in one of these cell lines, then we can create so-called cybrids or cytoplasmic hybrids, where we can create cell lines where the nucleus is from one original cell line and the mitochondria is from another, and therefore we can sort of control for the nuclear background and determine whether the mitochondrial mutations are actually contributing to the tumor. So I'll close there, and thank you for your attention, and I'll be glad to take any questions. Yes?
So when you survey across the different tumor types, there's some that are tumors that are more dependent on glycolysis compared to standard respiration. And do you see any different associations that way? I understand you have the associations for hypernucleus. Is there any, as you don't use mitochondria as much, do things go more haywire?
Yeah, we've been looking at that, and there's, initially there were some hints of those sorts of patterns, and we were actually really excited when we seemed to be seeing some hints, but that doesn't seem to be standing up to further scrutiny. So, that's certainly an attractive hypothesis, and we were chasing it, but unfortunately, it potentially does not seem to be globally panning out. Yes?
So for many of the mitochondrial diseases that are heteroplasmic situations, the mutations were found initially in the transfer RNAs. So I was wondering, but you seem to find fewer tRNA mutations. So I'm wondering whether you think that having a bit less of everything translated is compatible with is compatible in a neural tissue, while in the tumor tissue you can eliminate an entire complex and potentially make a useful intermediate that the tumor can feed on or whatever.
Yeah, it seems to be, we do seem to see that, and it's hard, I don't think, so a lot of these tRNA mutations that are important in mitochondrial disease are actually hitting the anticodon, and I don't think we've seen any evidence of that. So, my interpretation of that is just that, at least in the tumor cell, at least in the tumor types we've looked at, it doesn't seem to be able to tolerate that kind of disruption, at least not on a detectable level. I mean, one of the things is that although we have super high resolution in terms of depth of sequence, we don't have infinite resolution. So there could be some very low-level mitochondrial mutations that we're not detecting. Our level of detection is not infinite. So there could be some of these floating around, but in any case, they don't rise to high enough abundance for us to detect it.